So, good evening and welcome to you all. It's a great audience tonight. And it's my pleasure to introduce the speaker on behalf of the college. I'm the Dean for the College of Natural Sciences and Mathematics. And our invited speaker for the Spring 2018 Regenerative Medicine Lecture is Dr. Dustin Wakeman. Dr. Wakeman received his PhD from the University of California, San Diego, and then he joined the faculty of the Neurological Sciences Department at Rush University Medical Center in Chicago. In 2016, Dr. Wakeman joined the faculty of the Yale University School of Medicine, Department of Psychiatry, and he divides his time between his appointment at Yale and his position as a senior research scientist at RexGen Inc. And I just learned he's actually now living in Sacramento, so he's doing a lot of remote work from home, which he is enjoying. RexGen is a translational research company committed to advancing therapeutic discovery for human diseases by advancing the science of preclinical modeling to identify and select potentially effective therapies. Dr. Wakeman's research interests focus on determining the long-term value of using stem cell-based therapies for neurodevelopmental and neurodegenerative disorders, including Parkinson's disease, Huntington's disease, Alzheimer's disease, and other disorders of the central nervous system. In 2015, Dr. Wakeman received the prestigious Young Investigator Award for research he published in the Stem Cells Translational Medicine Journal in June of 2014, titled Human Neural Stem Cells Survive Long-Term in the Midbrain of Dopamine-Depleted Monkeys After GDNF Overexpression and Project Neurites Towards an Appropriate Target. This paper reported an important step in evaluating the efficacy of cell types and methods for stem cell therapies in Parkinson's disease. And this evening, Dr. Wakeman will be presenting a talk on stem cell therapy for Parkinson's disease. Please join me in welcoming Dr. Wakeman.
So thank you so much for that introduction, and I want to thank the organizers here at Sac State for inviting me to speak. This is a really exciting group of people to speak to, and I think, at least I hope that by the end of this hour, you'll all take away an appreciation for the amount of work that's gone in to where we are now. 15, 20 years later, in my case, where we've been trying to bring stem cell-based therapeutics to the clinic in Parkinson's disease. So I do have one disclosure. I was a consultant for Cellular Dynamics International and did receive funding from them for some of the work I'll present today. So now that we have that out of the way, let's talk some science. So I have 3 learning objectives for today. First and foremost, I hope by the end of this talk you'll be able to understand what these different cell-based therapeutics are and have been over the last 10 or 20 years for PD. Second, I'm going to try and define for you what I would consider to be an authentic midbrain lineage dopamine neuron and how that's changed over time. And then third, hopefully we can identify some of those key components that I see, at least, to establishing a pluripotent stem cell-based therapeutic for Parkinson's disease. So this is a diverse crowd, so I think I'll just start by defining what is Parkinson's disease after all. Well, pathologically speaking, Parkinson's disease is the specific degeneration of these neurons that are located within the substantia nigra you can see here in a patient. And what's important here is to understand that these cell bodies are actually located deep in the midbrain, sort of back here if you can imagine in your head. And they actually have to project fibers all the way up to the front of your brain, and that's where they release dopamine. And it's when these fibers and these cells degenerate that we lose dopaminergic transmission and subsequently get those clinical symptoms that many of you know, the tremor and the rigidity and the postural instability, at least the motor symptoms that I've just talked about. So the second major aspect of this study obviously is going to be stem cells. So I just want to, this very rudimentary slide, describe a few of the cell types that I'm going to talk about. First, we have the pluripotent stem cells, the embryonic as well as induced pluripotent stem cells. And then if we take those cells and what we call neuralize them or push them towards the central nervous system lineage, we get what's a multipotent cell or neural stem cell, so that can make astrocytes and neurons and glial cells. And then if you take those cells and you move them a little bit further along that dopaminergic lineage, you get something called a neuroblast or a full-on differentiated dopamine neuron. Now, the important part to highlight here for this talk is that these two cell types can self-renew. So we can take a cell population and we can make a whole bunch more of these. These two cell types, however, are postmitotic. So the number you have to start is the number you have for life, and if some die, you don't get them back. Now, cell-based therapeutics for Parkinson's disease is by no means a new concept. This has been going on for 100 plus years in some capacity. In fact, this cell type that are called fetal ventral mesencephaline cells have actually made it into clinical trials over several decades ago and have had variable results, some positive, some side effects. But the part I want you to understand about this is where the cell comes from. It's the part of the brain in a fetus that would eventually become the dopamine neurons that are going to degenerate in Parkinson's disease, okay? So everything I'm going to tell you about today and everything we're doing right now is to try and recapitulate this cell type in the lab. In other words, take what we know about normal human development and apply that to the cells we have to try and generate something that looks as physiologically similar to this cell as possible. Now, I just showed you that diagram of the different cell types. The first thing we tried to do was go back and use dopaminergic neuroblasts several decades ago. And the reason that we were trying to do this, as you can see, is these cells are actually derived from fetal cadavers. And you can imagine that sourcing fetal cadaver tissue to treat this number of patients is going to be problematic from sourcing, let alone the ethical issues. So let's set that aside. And as scientists, we're trying to ask ourselves, how can we make more of that cell type? So when I first started transplanting cells in Parkinson's back in my first year of graduate school, we were using something called fetal human neural stem cells. Again, they were derived from fetuses, but we could actually create a large number of these cells, and remember, they self-renew, so we can make more. And I'm going to tell you today about some of that work that we did in primate models of Parkinson's disease. In the second part of my talk, I'm going to talk about using both iPS and ES cells and the progress we've made with these cell types. Just highlighted here in red here are the studies I'm going to talk about. I do think it's important to highlight, though, this is a very large field with many people contributing. And you can see just in 2017 and even this early into 2018, we're making great strides towards bringing this cell therapy to the clinic. But I want to take you back now to when I first started in grad school in UCSD, and I was fortunate enough to be able to work on this project. And what we did was we took those human fetal neural stem cells, we transplanted those into monkeys that had been given experimental Parkinson's disease, and then we asked the question, could they actually make these animals better? So could they ameliorate the functional deficits? And sure enough, when we compared these to a sham control, we did find that functional deficits, some of them at least, were restored in these animals following transplantation. And when we looked at the brains, we actually found these cells that are called, these are Th positive cells that were donor derived. So for the rest of this talk, when I say Th, what I'm talking about is tyrosine hydroxylase. It's the rate limiting enzyme in dopamine synthesis. So anytime I say Th from now on, I'm talking about dopamine neurons, okay? So we actually went in and we counted these cells to see how many of them had become dopaminergic, and it turns out about 1% of them had. That was a little disappointing, if you might imagine, right? And so we started to say, well, could this behavioral rescue be because of this small number of cells? And we started to say, maybe not. And it turns out we followed this up, and we did show, in fact, that the majority of these cells actually remain as undifferentiated stem cells. What they do is they provide secondary trophic support to the cells that are remaining in the brain, to these dopamine neurons that are remaining in the brain, and that's really what's responsible. Unfortunate part was that it's not a long-term effect, okay? So we sort of said, okay, how can we enhance this effect? And we sort of turned to our colleagues at the time that were working on gene therapy, and we said, well, what if we marry the two? And so that's exactly what we did. We took this trophic molecule called GDNF, glial-derived neurotrophic factor, and you don't need to know what it is or what it does. I'm just going to give you the idea. It's like Gatorade for dopamine neurons, right? They really like this stuff. They need it. And so what we thought was, if we overexpress that protein here, that these cells might differentiate even more, and they might actually grow towards this target. Well, we looked at these brains a year later. We had to wait a year just to find out that, yeah, the GDNF was working. It was on. The cells grafted. They actually were maturing. They had these really nice fibers. And sure enough, some of them were dopaminergic. Unfortunately, when I counted them, 1%, no effect. So what we said was, let's take a step back, let's look at this. Is this really a cell type we think we're going to be able to take forward? And we said, you know, I think this might not be the best cell type. And so we decided that this was not going to be a good candidate. Now, why am I telling you about something that didn't work? This is an important story, and the reason is because right now, and for several years, there are clinics around the world that will offer you this exact cell type as a therapy for Parkinson's disease, and we know it doesn't work. We call them snake oil clinics or stem cell tourism clinics, and it's important that everyone, the public involved, patients, patient advocates, understand that this type of therapy is not ready yet for the clinic. And I'm going to show you now in the next 30 or 40 minutes just exactly where we need to get to and where we're at right now. So the next thing we did was we said, okay, what about human embryonic stem cells? Embryonic stem cells were starting to come of age at this point. We had a good idea how to turn them into neuronal cells, and we had techniques now to turn them into specific cells, specifically dopamine cells. Now, I don't want you to worry about any of, this is all published if you want to go back and look at it. Don't worry about any of the specifics that we actually do. You can look that up if you want, if you're really interested. What I want you to understand is that as stem cell biologists, all we're trying to do is recapitulate normal human development. This is probably why some of the best stem cell biologists in the world are developmental biologists, because they understand what it takes to make the human body. And so all we're trying to do is mimic what's going on during normal development. And so we can add these factors, and in this case, we created what are called neural rosettes. So these are these neural stem cells. We then added a variety of maturation factors, and we can make these mature neuronal type cells, a small percentage, 17% of which are actually dopaminergic. And they also have a gene expression profile similar to dopamine neurons, and they also secrete dopamine. So we kind of looked at this data and we said, well, it looks like we got the right cell type. They're making dopamine. Let's give these cells a shot. Maybe we'll have a better chance. So that's what we did. We injected these cells into the same monkey model of Parkinson's disease. And sure enough, we got these beautiful grafts here in the striatum, as well as in the substantia nigra. And we labeled them with this GFP protein so they would fluoresce green so we could find them. And you can see, it's kind of a little bit difficult here to see, but they had really nice fibers and they looked like they were mature. Well, then I went and stained them with tyrosine hydroxylase, that dopaminergic marker, and sure enough, 0% of them were TH positive. So throwing my hands up in the air now, right? What gives, right? Well, it turns out we were just a few steps too late with our timing on the developmental cues. And I mentioned that the developmental biologists are the ones that really understand what's going on in these systems. And a very talented developmental biologist by the name of Ram Mackay, a gentleman, a postdoc in his lab at the time, published this paper in 2007. And what they showed, now this is a mouse brain, but what they showed is that these dopamine cells, the very first ones actually arrive from this area called the floor plate. And so turns out you need floor plate cells to make dopamine cells, midbrain dopamine cells. And we were making what are called neuroepithelial cells. These are the wrong cells, okay? Well, another guy was paying attention to this, a guy by the name of Lorenz Studer. He happened to have done a postdoc with Ram Mackay and has been working on this for about 20 years before I came along. And what Lorenz did, along with Sonia Cricks in this paper that was published in 2011 in Nature, was they showed that this rosette-based patterning we were doing was completely wrong. We were completely off. And what we had wrong was we had the timing wrong. And again, the specifics don't matter here. We just weren't making the right cell type. And so this paper was a paradigm shift for our field. Everything that had been done before was kind of thrown away. And I'm telling you, it was overnight. It was within a week or two, every lab in the world had switched to doing this. Now, myself and Jeff Cordover, who I was working with at the time, were fortunate enough to be able to work with Dr. Studer on this. And I'm going to take you through just some key pieces of data from this because they're going to set up for what I talked about earlier in defining what a true midbrain dopamine neuron is. So these cells express these transcription factors up here. You see THs up there, and they express these other transcription factors, FOXA2, which is a key master regulator, which Mackay figured out. And you can see when we use their method, we actually get large percentage of these cells that are positive for these markers. Whereas our other technique, yes, we made TH cells, like I did in that paper I showed you that didn't work. Unfortunately, we didn't have the right cocktail. Okay, so not only are they making the right cells, but they're not making the wrong cells, which are these serotonergic and GABAergic neurons, a different type of neuron. So Studer took these cells, he knew he was onto something, and he transplanted them into a rodent model of Parkinson's disease. This is 6-hydroxydopamine. It creates a lesion primarily specifically of the dopamine neurons. In this case, it was a mouse and a rat he used. And he transplanted those cells into the brain and waited 5 months, and sure enough, 5 months later, those cells were Th positive, as well as positive for these other sort of what we call master regulator genes. Well, these graphs are beautiful, and those pictures are beautiful, and they'll probably get you the cover of a journal somewhere. But at the end of the day, I'm not into journals. I'm into trying to see if we can come up with therapies. That's what we're all trying to do. So did the animals actually get better? That's the question. And it turns out they did. By 3 different tests here you're looking at, the floor plate-derived cells actually reversed motor deficits in these animals. And importantly, by 2 pharmacologically independent means, which are much more difficult to alter. So this is actually the point when Dr. Studer and Dr. Cricks contacted myself and Jeff Cordova at Rush, and they knew that we had a history of transplanting cells in Parkinson's disease models, and they said, would you be interested in working with us? And I said, I remember Jeff came to me and he said, do you know who this guy is? And I said, yeah, he only publishes in Cell, Nature, and Science, and usually Nature. We're working with him. And that turned out to be a really, really good decision. So what we did was we took these cells and we transplanted them into a Parkinsonian monkey. I'm going to tell you more about that model later. But what I want you to understand, this is 3 months post-transplantation. This brown stain here, these are human cells that were put into the monkey brain. So anywhere you see brown there, those were the donor cells. And I showed you that I was able to do that before in the paper where we failed. What we weren't able to do is keep these cells dopaminergic. So the critical question for me was, were they dopaminergic? And you can imagine how happy I was to find all these Th positive cells in here. And not only were they Th positive, they were FOXA2 positive, that master regulator. And that really is why this worked, because they were patterned correctly. And we took a look closer at those neurons. They have this beautiful mature neuronal morphology. They look like textbook dopamine neurons, these angular somata. And to put this all into perspective, since I know a lot of you don't know the pathology of this disease, remember at the beginning when I told you there were clinical trials using those fetal cells? This is actually a piece of tissue from one of those patients' brain from an autopsy that we stained with Th. So this is what those cells look like. This patient lived for 10 plus years with these cells. And this is how they compare to those cells I just showed you. You see the morphology is very similar. They're not quite as elaborate as those. This is only 3 months out, and human cells, as we know now, take a long time to mature. So I'm ecstatic at this point. I'm junior faculty now at Rush, and I've got really exciting data, hot data. And so the next thing we started to say was, well, let's think about the clinic. And what do we know about stem cells that might be a deterrent in the clinic? Well, stem cells, as I told you originally, they self-renew, right? Do we know other types of cells that self-renew? Cancer cells self-renew, right? So the last thing I want to do is put cells into you and you get a brain tumor. So what we have to be really careful with is making sure that we don't have cells that are aberrantly growing, or at least are slowing down their growth. So we did a stain, it's called Ki67, that doesn't matter. The point is, where you see a black dot, that's a cell that's actually proliferating, it's growing, it's replicating. And we never saw any real, you saw a couple little groups here, but over time you can see this number diminishes. But what it told us is, 1, we're on the right track, but 2, we still have work to do. How do we get rid of these cells? How do we purify this population? You can always make iterations on your product. This is all just basic research product, proof of concept. Now, I'm going to pivot there real quickly because I want to tell you, we obviously followed this 3 months up, and we did a 1-year-long study. And it turns out those cells survive for 1 year in the monkey brain, and they also continue to express tyrosine hydroxylase. So we'll come back to this at the end of the talk, but I want to just quickly summarize for you is Studer and Cricks basically figured out how to take a pluripotent cell that you can make as many as you want, scale that up, and make real dopamine neurons, midbrain dopamine neurons that I was telling you about, and then they tested those in animal models. Now, we were a little concerned with those other cells, obviously. So there are a number of different ways that we've looked at trying to get rid of those cells or altering the composition. I'm just going to tell you about one just so you get an idea of some of the things that are going on. So this is called sorting or purifying the cells, and in this case we just used a magnetic antibody, so it's an antibody that has a magnetic bead that attaches to it, and then you can pull cells out. And we compared these to non-sorted cells. This is 6 months post-transplantation in what's called a nude rat, so they're athymic, and all that means is that part of their immune system is gone, and the reason we do that is so they don't attack the cells because they might see it as foreign, much like you would if you got a heart transplant and you might reject it. It's very similar. So you can see these grafts survive. They have these beautiful fibers that are growing out from them. Beautiful TH fiber, or TH positive neurons. There was actually no difference between them. So this is actually an example of where we had a really good idea and there was some really great data to support it, but it didn't really pan out in the long run. Okay, which turns out how a lot of science works. I'll give you guys the in on that. A lot of science doesn't work if you didn't know that already. All right, so but what did we learn from this? Well, I'll tell you what we learned. We learned a lot. We learned that these cells actually mature a lot over the first few months, but that it actually takes a long time for them to really get to where we want them to be. So this is just 2 weeks later. This is what we call an immature bipolar morphology. Point being, we go 6 months later, you see that nice angular morphology, you see fibers everywhere. This is in a brain that is not dopamine deficient. This is like a normal animal. This is just a proof of concept. So we then looked at what it would look like in the dopamine deficient brain, the 6-hydroxydopamine brain. And sure enough, we got these beautiful graphs, and they were also positive for, this is human NCAM. Again, it's a human-specific neural marker. So anywhere you see that stain, that's a human cell that we put in there. And so you can see these cells are co-positive for this tyrosine hydroxylase. They're also FOXA2 positive, remember, that master regulator I told you about. And in Parkinson's disease, there are, the area that's most affected for the motor disorders, there's 2 different types of dopamine neurons, A9s and A10s. A9s are predominantly destroyed and responsible for those motor movements, and so that's predominantly what we're looking to replace. And sure enough, most of these cells are of this A9 origin, which are they're positive for this protein called GERK2. And of course, there were some that were of A10 origin, as we expected. Now, what we did not expect at all was that when we put these cells up here in the striatum, that they would actually project all the way back into the substantia nigra, kind of a reverse of what normally would happen. But actually, you can see here, already at 6 weeks, it's hard to imagine unless you're sort of in this field, but that is an incredibly long distance for a fiber to grow through the brain in an adult brain. So we knew we were onto something here, and in fact, it turns out that these fibers in here were actually dopaminergic as well, so they're actually following the normal nigrostriatal pathway. And you can just see down here, there were various types of fibers, but the point here is that they were maturing. Now, what has happened with all this? I just showed you a very, very small portion of the data and the small portion of papers that have gone towards this work that Dr. Studer started. Last year, Dr. Studer and his colleagues at Sloan Kettering in New York founded a company called Blue Rock Therapeutics that raised a record amount of money on their first offering. And they're now performing their late-stage IND-enabling safety studies. So for the patients out there, this is huge. This could be the very first product in the United States that offers hope 10, 15 years via a cell for Parkinson's disease. They're getting very close. They're doing their diligence with safety. They're doing it the right way. And that is critical, doing it the right way. So now, the second half of my talk, I'm going to pivot to another type of cell, the iPS cell, pluripotent stem cell, and work that I've been doing with a company called Cellular Dynamics. And the reason, one of the things I need to highlight here is the immune system. This system right here using embryonic stem cells would be equivalent to grafting, in most cases, giving you a heart or a liver transplant, but not knowing if you're going to reject it or not. So we have to give you drugs so it doesn't reject it. That's a simplified version. Or you go through a process that you would normally go through if, say, you were on an organ donor transplant, and you do what's called HLA matching. You're matching genes that are responsible for the immune response. And the reason that iPS cells offer so much promise is because theoretically you could take the cells from you, make them into dopamine neurons, and give them back to you. And you should, at least theoretically, not need these drugs to maintain over life. So it offered either a way to do the graft yourself, or you can imagine if we created a lot of different versions of these from people with different genetic backgrounds, we could then match you to that background. So we could take you out of a bank and match you to that background. So the rest of this talk now, I'm going to be talking about this work I did with Cellular Dynamics International, who recently was bought by, I guess not recently, about a year or two ago, was bought by Fujifilm to develop therapeutics. And this is my basic stem cell slide. Many of you have seen some version of this. Mine's pretty dumbed down, but I think it drives the point home. The idea is you have a patient. In this case, we took blood cells from them and we put episomal transcription factors in, created iPS cells. Now, classically, and what this company I was working with was doing was they can then take those cells and make pretty much at this point any cell type in your body, okay? In this case, we're making dopamine neurons. But you can take those, and one route is use them as a research tool to do drug screening or do disease modeling, try and learn more about the disease or how to target certain drugs. We took a different route. We were going for cellular therapies or cellular transplantation. Now, this is the autologous route I told you about. That would be taking the cells from you, the blood from you, giving them back to you. This other route would be this, what I call this haplomatching approach, which is the approach that the company has now decided to take. Now, why am I putting a picture of a vial of cells on the screen? Seems like a pretty bizarre thing to do, but this actually holds the promise, in my opinion, for creating a technology and a therapeutic to reach a very large number of people all over the world. Because the FDA and other regulatory agencies, if you're going to try and put a drug or a cell into somebody, you need to make sure it's safe, and it has to go through a whole set of criteria, and they have to be characterized. It takes a lot of time, it takes a lot of money, and you would need to do this every single time you created a new batch of this. I don't care if it's a drug or what, that's the way it works, right? And so you can imagine we have our scientists in the lab, and they spend 7 days a week, 8 to 12 hours a day in that lab culturing cells. And it's a stochastic event. It's not the same every time. And so it's very hard to control. And so what this does, by being able to make large amounts of cells and freeze them, you can do all your characterization. You can create a huge lot. You can then disperse that, right? You can ship frozen cells around the world. And then the final piece that makes this so important is that imagine yourself in reality, and you're a neurosurgeon. And you call down to the lab and you say, in 1 hour, I need 20 million cells. I need 100 cc's of cells. And I need them in an hour, because I have a patient I'm doing surgery on, deep brain surgery on. And there's a technician there, and they're working diligently, and then some cells aren't good, and some cells are, and this goes on for 2, 3, 4 hours. And we're introducing human error. That is not something that we can scale up to meet a large population of people. So it's critical that we came up with techniques to freeze this. And when I started this work, why I'm belaboring this point is that when I started this work, this did not exist. You could not take a postmitotic dopamine neuron and freeze it and then thaw it back, and it wouldn't. This didn't happen. And in fact, when I started talking to this company, I told them, I don't believe you, because this isn't possible. And they said, all right, we're going to call our bluff. Let's work together. And that turned out to be, another really good decision. So this is very straightforward what we decided to do. We said, we're going to take your cells out of the vial, we're going to thaw them, we're going to rinse them real quick, and we're going to transplant them. Again, this couldn't be done. And all the work I'm going to show you now is primarily 95%, maybe 5% was me, but 95% of this was two very talented grad students, Benjamin Hiller and David Marmion, who are now finishing up their PhDs at Rush with Jeffrey Cordova. And the question was simple. Can we transplant these into animal models of movement disorders, in this case Parkinson's disease, and will they function? So now I'm going to show you a whole bunch of data, and I'm going to run through it a little bit fast. It's all been published. It's all open access. But perhaps one of the most important pieces of data I'm going to show you is actually this graph right here. And it's really boring, actually. But what this shows, this is 16 different production lots of those cells, those frozen cells. And it shows that you can thaw them, and 70% of those cells survive. All right? This was the proof in the pudding for me. So the next question we said was, OK, your cells survive, but what are they? I got to convince myself that these are dopamine neurons, the real thing. So we did a variety of tests. We looked at gene expression profiling. We looked at the protein expression. And again, don't worry about the specifics. I'm happy to talk to you later. You can look it up. The point is they had all these characteristic genes and proteins that we come to expect during normal midbrain dopamine development. You remember that FOXA2 and that TH. So then we said, OK, great, they look right. What about that safety aspect, right? What about the proliferating cells? These are two markers. There's that Ki67 proliferative marker, and here's EDU. These are just two ways to look to see if your cells are dividing. Less than 0.1% of those cells are actually dividing. So these are really our postmitotic, or remember, what you have is what you get, cells. The next question I wanted to know is, do they function, right? They make pretty cells and they make pretty graphs, but do they actually function? And before we did that in animals, we wanted to see if they could do it in the lab. So we looked, and sure enough, they can both make dopamine and secrete it when challenged with potassium chloride. And they also fire action potentials. Now, this is a very small portion of the electrophysiology work we did in collaboration with Jim Surmeyer at Northwestern, who knows much more about this. So I'll tell you as much as I know besides these are squiggly lines, and that is that they will fire both spontaneous and evoked potentials if you give them a current, and they also exhibit dose-dependent inhibition of both sodium and potassium channels, which is indicative of a functional ion channel and a functional neuron. Again, we did a whole bunch more here to show that they fire similar to midbrain dopamine neurons, but we don't really have time for that. Let's get to the point. Let's transplant these cells, right? So, what's our model? I want to just explain the 6-hydroxydopamine a little bit more in detail. You take the 6-hydroxydopamine, you inject it into the brain in this very specialized structure called the medial forebrain bundle. That's a nice way of saying the fibers that run from where the cell bodies here are in the substantia nigra up to the striatum. And the important part here is stringency of our testing. I won't go into the details, but this is the substantia nigra back here, remember? And this is the side that hasn't been treated with a toxin. So those are your healthy ones. And you can see they're gone here. And you can see this is the side that was treated over here. And you can see all those fibers that went to the striatum, to the front of your brain, they're gone. This is the control side. They're all still there. Why is that important? Because if we see behavioral improvement in these animals, it's because of the cells. It's not because these cells grew back or maybe they were kind of dead. It's because the cells were actually working. And I'm going to show you evidence for that. Very straightforward. We took these cells, we thawed them, we spun them down twice. It takes 22 to 25 minutes to do this process. Very simple, very straightforward. Any laboratory technician in any hospital that has experience with this type of work could easily do this. And that was what we were going for. What is the ideal scenario? We can backtrack off that later and fine-tune it if we need to. So that's exactly what we did. This is 6 months later. This is the side that we injected. Those are the TH-positive dopamine neurons, and they are innervating the entire brain. We're going to come back to this picture in a minute. Important part here, remember that substantia nigra I showed you earlier? It's still gone, okay? So again, we didn't rescue these cells like we did in that very first paper I told you about. We didn't suddenly get sprouting from those cells. If we see functional improvement, it's from these cells. And so that's that same image I'm showing you. Now I've inverted it, turned it dark field so you can see it easier. And you can see this entire structure here is innervated. And this area is the striatum. This is where that dopamine needs to release, okay? And sure enough, they're human NCAM positive, so these are from the grafts. These aren't endogenous fibers that are already there in the brain. These are actually what we transplanted. And we quantified that, and we showed that they were statistically significant. Don't worry, we cut this piece out. We call that cherry picking. We would have had significance with that, but we wanted to make sure this was real. So indeed, we have a significant difference. We then wanted to look at those cells. What do they look like 6 months later? Extremely mature fibers coming out. You see the very large cell bodies. Again, very similar to what you would see in the actual brain itself. About 20 to 30% on average actually survive, and that's more than enough, it turns out, to function. Then we wanted to turn to safety. Remember, FDA, safety, safety, safety. So this is it's called human nuclei. Basically, where you see a little black dot there, that's a cell. Well, it turns out they all stayed where they were supposed to stay. They didn't migrate out, which was great for us because the FDA wants to know where do they go, and if they go other places, you need to show them where they go. Turns out they stay put. That's great for me. What about proliferation? This was shocking data to me because I had never seen this before in all the years of transplanting different cells. There's the graft right here, and there were 0 Ki67 positive cells in all but one animal. One animal we found like 1, maybe 2, and we think it was an artifact, but we counted it anyways because we wanted to be on the safe side. And we know that because there's actually an internal control in these animals. Turns out all of us actually are making new neurons, slower pace when we're adults, in this area right here called the subventricular zone. So we have a nice control there. So we know that these cells aren't proliferating. What kind of dopamine neurons were they making? I told you we needed FOXA2 on, right? There's FOXA2, TH. Boom, we hit that. What kind of TH cells? A9 subtype neurons? Yep, most of them are GERK2 positive. There were a few of these A10 type, but that's okay again. Okay, got a lot of pretty pictures up there. I'm feeling pretty good about maybe getting the journal cover, but does that matter? No, right? Do they work? That's what we're trying to do. That's the end game. Can we help people? And I wouldn't be here right now if they didn't work, obviously. So this blue line right here is animals that had a successful graft. And you can see, if you go down here, that's better, okay? So you want to get to 0. That's getting towards normal. And all the animals that had a successful graft actually had complete restoration. This test right here is kind of a test surrogate for the release of dopamine. And this test is a little bit more stringent, and it usually lags a little bit because that actually typically requires innervation. So this is really nice evidence, and this was trending towards becoming completely restored. And those data are actually in process right now. So we're 15 years out of a lot of failure, and we're finally to a point where I feel like we've got a pretty good cell type here. We've got the right functional response. You can probably imagine what comes next. We wanted to transplant those into a large animal model. So here's that model I was telling you about earlier. It's called MPTP. It's a selective neurotoxin. You can inject it, and it will kill the dopamine neurons in almost identical fashion that you would see in the human brain. It's very specific for these A9 neurons for some reason. We don't know why. Quite frankly, it doesn't really matter. The point is it's a great model. We call this a clinical phenocopy of the disease. You get the characteristic loss of dopamine neurons in the substantia nigra, so the cell bodies, as well as loss of the striatal transmission. Importantly, you also get the clinical manifestation. So you get the rigidity, you get the tremor, you get the postural instability and akinesia. So this is a really great model. So we wanted to see in the dopamine deficient brain, in other words, the environment that you would see in a diseased person, can these cells survive, or is it something about being in a healthy brain? And 1 month post-transplantation, sure enough, we had nice healthy grafts. They're starting to send fibers out into the host brain. They're kind of bipolar in morphology. They look a little immature. So we wanted to say, well, what happens a little bit later? So at 3 months, we looked at these brains. And it turns out, I hope you can appreciate the difference, highly, highly mature, have sending axons and fibers out all over the brain. They are TH positive, and they're homogeneously TH positive, whereas many times they only mature on these outside areas. And they also, of course, need to express FOXA2. They do, and GERK2 as well, that A9 subtype. And again, all this work has been published. It was published in last year, and open access, so you're all free to go check it out. If you'd like to repeat it, great. Let me know how it goes, because I promise you it's repeatable. We've done this now, I think, 3 or 4 times, and every time we get the same result. Okay, now, I want to sort of shift gears now, and I want to tell you about what's ongoing right now, and then what's in the future, and what the clinical timeline looks like right now. Oop, wrong way. All right, so everything I just showed you was using cells derived from a healthy human being. So how does that relate to the disease? Well, if you think of it, I talked about the matching, right? The HLA matching. So we could take a healthy human, get cells from them, make these cells, give them back to you if you have the right genes to that are compatible. That's that scenario. What about giving them back to you from you? Maybe there's something about the disease that changes the cells. So we have actually, in collaboration with Deborah Hall, who's a phenomenal movement disorders doctor at the Rush Movement Disorders Clinic, have actually isolated, taken blood, and derived these cells from sporadic PD patients. And we were very specific in choosing patients that we thought would have the same profile of what our ideal candidate might look like down the road. So planning ahead. Remember that, planning ahead. There's also a 9-month functional study that, again, in these athymic nude rats, so they don't have the immune system, so you don't have to worry about them getting rejected, where we're looking at synapses. So how the cells connect to the brain and how the brain connects, and see how that works. Because that's actually important, and it's very understudied. And then I'm going to show you next a slide. Now we're going to transfer into my current position at RexGen, and we're going to see how we've taken this now from a research grade academic project into a clinical path, and starting to look at how are we going to do this and how are we going to put this together for the clinic, and what's that look like. So I'm going to show you data, safety and tox data, basically redoing the study we just did with a cell type that is made for the clinic. Everything I just showed you was called a research grade cell. I'll talk a little bit more about that later. But the important part is that the therapeutic you're going to use in the clinic has to go through all of these tests all over again. So everything I just showed you was proof of concept. It has to be redone. And then finally, what's underway right now is what's called a good laboratory practice, or GLP, safety tox study. And this is to show that your cells are safe. There's a number of parameters at the FDA, and each program's a little bit different. But you have to go through this step. It's extremely expensive. It takes a long time. All right? So again, it comes down to planning. We're going to get to that in just a minute. First thing I want to do is I want to show you when we redid that same study I just showed you, that 3-month non-human primate study, MPTP monkeys, with my company I'm at now, RexGen. So we are a disease modeling company. We provide disease models for CNS diseases like Parkinson's or eye diseases, wet macular degeneration, aged macular. So what we did was exactly the same. And what you can see 3 months later, this is the clinical grade cell now. This is actually, they've refined their techniques. Remember, I talked about we're always refining techniques. We're actually getting better graft survival, better outgrowth of these cells, and they're also TH positive. So this now is proof of concept going forward towards your, what we call your IND package, or your set of data that you're going to go towards for the FDA for safety. Okay. So the last 2 slides here, I'm just going to kind of try and bring this all together. And I told you that last learning objective is, what do I see the path forward, and what do we still need to do towards bringing this to the clinic? Because we're still not quite there yet. Now, this is actually turns out a very simplified graph. This was actually by Lorenz Studer and Vivian Tabar, who were running that Blue Rock Therapeutics program that I told you about. This was published several years ago now. But what I want you to first notice is that this is a sort of a dumbed down version of the path to the clinic. That is not a straight line. That is a cyclical process. And what you need to think about when you're, especially think of you students out there, or even your professors out there, is you're thinking about envisioning what your final product might look like, or where you might want to take your new drug or your new cell type. You actually, it turns out, you have to start planning in reverse. You actually need to kind of think about what your clinical trial is going to look like, figure out how you're going to validate that and get it through the FDA, and then make sure that you're planning all your small animal studies. Because as I just told you, if you start with the wrong cell type or you tweak it a little, guess what? You get to go back to go, and you don't get to collect $200 on the way. In fact, you get to spend another $10 million maybe. So it's really important, and you can see that these, again, you could start your phase 1 clinical trial, and you might have something that goes a little weird. You have to start, kind of go back, redirect, recalibrate. So it's really important to be rigorous with your planning, okay? And this is an extremely dynamic process. We're all very fortunate here to be in Sacramento at UC Davis, who has a phenomenal regenerative medicine program and has the capacity to generate GMP quality cells. And they're doing this actually, and they have therapeutics that are in the clinic, oncology candidates, and others here in the audience that are working on Huntington's disease and other rare diseases. Okay, so this is an amazing place we're at right now that has all these components put together under one area. So I think in my last slide here, I just want to, second to last slide, sorry, my last data slide, I just want to bring this all together for you. Where are we at? What have we done? So I told you about the work we did, started with Lorenz Studer and Sonia Cricks, where they had this paradigm shift, this just amazing transformation in how we approached making the cell, defining the cell. They've now taken that in just tedious amounts of work, have now formed Blue Rock Therapeutics, and are doing these late-stage studies, are developing their therapeutic for the, and I do believe that they will be filing for IND soon. So I think you'll see your first clinical trial 2019, maybe at the earliest, late 2018. Their cells are all ready. It's a very exciting time. You can tell it's an exciting time for Blue Rock because they raised a lot of money in their first round of funding. In fact, they broke a record. That's using embryonic stem cells. Now, I told you at the beginning, all we're trying to do is remake this cell type, the fetal ventral mesencephalic cell, right? This little area in that brain that has the dopamine neurons. That's what we're trying to do here. We're trying to make those. We have iPS cells here. We're trying to make those. And so there were clinical trials in the past using these fetal cells. Turns out there's actually one ongoing right now in Europe by a group called TransEuro. And no, they're not envisioning treating every Parkinson's disease patient with all this tissue, but they are looking at this and saying, hey, we've got a lot of information from the system. Let's learn how to refine these techniques, and then we're going to apply that to these cells, right? So taking all the body of knowledge and bringing it together. This is you have a group, a huge group in Europe. We also have a group in Lund in Sweden, Malin Parmar and Agnet Kirkeby, that are developing a very similar product to Lorenz Studer. So we have now, we have Sweden, we have, this is Cardiff, basically. We have New York via Boston now. I told you about Cellular Dynamics, which is in Wisconsin, and is also now owned by Fuji, so a Japanese entity as well. There's also an amazing, amazing neurosurgeon and developmental biologist, Jun Takahashi in Japan, that has been doing unbelievable work 2-3 year long experiments in non-human primates showing that his cells, which are very similar, truth be told, big secret, all these cells are pretty similar to each other. They have little fine-tuned trade secrets, but they're all pretty similar, which is good for all of us because they're all very careful. All these groups actually work together in something they call the PDG force. So all the people that are, quote, competing are actually working together to try and bring real clinical value to you and bring a good, safe product to you. Now, you might ask, just like I did, well, which one's the best? The answer is, I don't have a clue. I hope they all work, okay? But the point is that they all need to be tested side by side rigorously, and they all need to have a chance, okay? So you can see we have 3 very different ways to hopefully come up with the same thing. But at the end of the day, students, listen, you're not treating animals. You're treating patients. You're trying to bring new therapeutics to patients. And in Parkinson's disease, we have a couple of really good options. We have oral L-DOPA, and we have deep brain stimulation, which have very, very good efficacy, maybe, albeit 8-10 years, maybe a little bit less in some cases, a little bit longer in other cases. But if you're going to tell me that you're going to stick a living cell in my brain, and I'm going to take a risk, and you're going to do a surgery, and I have the option of a pill or an adjustable device that I can turn on and off, what are you going to choose? So I posit this. For these therapies to be useful, to be widespread, we're going to meet, my personal opinions is that we're going to require at least a 1- or 2-fold enhanced efficacy over the currently available treatments. If you're coming up with therapeutics that are as good as what's on the market, you need to try harder. You need to reformulate, okay? We need to improve quality of life in patients. And in Parkinson's disease, you're talking 10, 15, maybe 20 years out. So I hope what I've been able to do for you all here is sort of paint a picture, a very broad picture through my experience of trial and error, steadfast science, reconfiguring the way you think about something you think you have locked down, a la how we actually make these cells, and then the type of efforts, group efforts, through academia that require industry working together to bring something to the market. And at the end of the day, we need that therapeutic to be better than what's already on the market. So you students out there, you guys that we met earlier, be thinking about that. How am I going to treat the patient? What are the symptoms that I'm treating? These are motor symptoms I was just talking about that we can train. We can fix. We can ameliorate with stem cells. Choose my words carefully there. There's a whole entire understudied area of Parkinson's disease, later stage Parkinson's disease, that is completely neglected for the most part. And in my opinion, that needs to be, the next frontier. So if we can make these cells and we can replace your dopamine for 10 or 15 years, and in that 10 or 15 years, we can come up with ways to modify the disease, to actually stop the disease and the autonomic function so that you're not in a wheelchair, you're not aspirating, so your heart works correctly. Then I think we're going to have a real shot with this disease. But it takes steps. It takes a lot of people working on it. So maybe you're going to have the next big idea, right? You never know. Constantly be thinking, be working, and as we talked about, students, network, talk to people, get to know people. All right? So again, I want to thank the organizers of this lecture for letting me speak. I hope I've imparted you guys, where we're at now, some of the roadblocks we've encountered, and what I see as a very, very promising next 5 years in this field. So with that, I just need to have my obligatory acknowledgement slide. There were a whole lot more people than this involved in all these projects. I want to name a few by name. Jeff Cordover, who took a shot on a naive grad student back a long time ago, back in Rush University Medical Center. My 2 amazing grad students, David and Benjamin, who really are just going to be phenomenal scientists here soon. Cellular Dynamics, Chris McMahon is the guy that runs their dopamine project there. Carter Cliff's the guy that called my bluff when I told him you were full of it. He said, try it out. And then, of course, Lorenz Studer, Sonia Cricks, who without them, this wouldn't exist. I wouldn't be up here right now talking. And then all the other people along the way, Deborah Hall, who's just a phenomenal physician, and taught me how to look at human disease for what it was, human disease and not an animal model. So thank you very much for your time, and I'd love to take questions.
Do we have any questions in the audience? I have a microphone so we can actually hear you. All right, I'm gonna.
How are you doing?
Good, how are you?
Good. So the field of study that I'm interested in is comparative research between things like Alzheimer's and different neurodegenerative diseases. And I'm curious, any of the findings that you found for this disease, does it carry over to other neurodegenerative diseases?
Yeah, that's a great question. The simple answer is yes. A lot of it carries over. We can apply the same principles that we learned here. We can learn about, developmental timing of cells. Turns out, when you don't make the dopamine cell, you make another cell type. That's a different disease. That's something that somebody will pick up, and they do oftentimes pick up. Cell replacement strategies are definitely amenable to certain diseases. Parkinson's disease is an ideal candidate because at least the motor symptoms and the majority of the degeneration in the brain early on is in this specific cell, a very specific cell with a very specific target. And in fact, you notice that we were putting the cells in the striatum, not in the substantia nigra where they normally are. That's because actually all you need to do is get the dopamine to release in the right area. If you think about taking a pill, it's just available everywhere, right? So as long as it's hitting the area it needs to be in. The other reason is if you stick them in the substantia nigra in a human being, we learned this through the clinical trials, they don't work. Very big difference between a rodent and a human scale of the brain. I talked about how remarkable it was that those fibers grew that distance in 6 weeks. You can't even see what that is, right? Multiply that, 10 or 20 at least, just to get to the beginning structure you would need to get at. So there's anatomical reasons. There's a whole set of patient selection criteria that you would want to apply to that. But yes, the bottom line is you can apply these same principles to other diseases. You just have to understand again, what I was talking about, understanding your disease, understanding the symptoms of the disease that you're trying to target, and just understanding your test system, I think, is the most important part. Alzheimer's disease is a little bit different because it's a lot more globally spread, obviously. So I'm not sure I would want to tackle that right away with this type of therapeutic.
So you were saying the treatment need to be more efficient?
Pardon?
So you said the new treatment need to be more efficient. So how about the cost? What does it cost?
Cost. Oh. Yeah. Okay. No. The reality is that an extreme amount of money goes into research and development by the pharmaceutical industry, by the academic, I don't care who does it. And there has to be a recoup on that cost at the drug level, right? And we as patients or consumers don't always like that. We struggle with our insurance companies. I don't know, you have to get to the late stage when it actually becomes the drug goes to market, and then that's a negotiated rate. Actually, we don't have time to go into it, but it's much more scientific than you would imagine, and it has to do with how much you can recoup from the cost you put in and how long. Hopefully, a therapeutic like this is going to be a one-and-done situation where we do it once and you get better. The reason that sometimes pills are drug companies like pills is because you have to keep buying them, and you can keep making money off them. But I don't think. I think it's a misconception that pharmaceutical companies jack the rate of drugs up so they can just gouge you. It's not true. I mean, certainly they're a business, they need to make money. I don't know what this therapeutic is going to cost. I know it's not going to be cheap to start. I can tell you this. The first iPS-based cell therapy was performed in the eye for macular degeneration. The Japanese did it. and they were going to do everything with autologous cell types. So your cell type, they took a patient's cells, they turned them into the specific kind of eye cells that were being degenerated, and they transplanted them in. They did one patient, and they realized that there's no way that they're going to be able to fund this, as a full therapeutic. This is actually why CDI is going with the haplomatching. So you can imagine if you invest money in the front on a number of cells that span, and they've done this actually with CIRM support, actually, California Institute for Regenerative Medicine, everybody out there. If you're not familiar with who they are, please support them, continue to support them. A lot of the people that are in this room today are supported by them, and the good science happens because of them. The CDI has gone with the haplo approach too, because you can just make a lot of cells, and then you just go in and you know the genes of those cells, and then you can just genotype or look at the genes of the patient and try and match them. Again, I can say that very simply here, but it's a big endeavor, right? You have to start somewhere. Do I think autologous therapy will eventually make it? Yeah, I mean, I think the FDA will eventually get to a point where they'll bring the regulations down, but that's only going to be through proof of concept and showing that every single time you do this, it's a safe product. So if you're going to, just to give you guys an idea, for those that aren't aware, so if I were to take your cells and pull them out of you and want to put them back into you, I have to go through that entire process for each and every one of you, which is going to be a 5 to 7 year process maybe. Let's say we really get good at it and it's 1 or 2 years. You have Parkinson's disease, you have a little bit of time to wait. What's it going to cost then? Because now you're paying for the full price of the therapeutic in one, and I can tell you it's just not feasible as of today. That being said, I was sharing with the students, I remember when I heard first when the iPS cells, these cells that you could take from your skin and turn into any cell type of your body, and we all said that's bogus, that's not real. We were wrong. You know, technology is moving at an incredible pace. The throughput is amazing these days. So do I think we'll get there? Yeah, probably. Will we need to? I don't know. We'll see what happens along the way. But I do know that for Parkinson's, you will see clinical trials starting in the next 2 to 5 years. Looks like we're over there with the mic. We'll come back. He's got the mic real quick, then we'll come back.
Considering that Parkinson's is a time-sensitive issue, how does the timeline of this regenerative therapy compare to the timeline of the disease progression?
Okay, so what I think you're asking is, when would you want to intervene with the disease, considering you're on a spectrum of degeneration and you're getting worse over time?
And how long it takes to produce these neurons?
Great question. So we know empirically from the fetal cells that they start to be optimally working around 2 to 3 years is when you really start seeing therapeutic benefit. So again, you can take your pill and get immediate effect, or wait 2 to 3 years until you really have something working. The way the FDA is set up right now, and appropriately for safety, is they're not going to let you go into an early stage disease. Okay, one thing I didn't really hit on there was, imagine if you had a few cells that were growing, a few of those proliferative cells. Let's say it's ALS. ALS is pretty steep decline. Parkinson's, you got good years of life left when you're diagnosed. And most people, unless you have a really aggressive form, are going to get 10, 15 years, give or take. So there's a lot of time for those cells to grow, which would be catastrophic. It would, this research would basically end. So the patients will probably going to be allowing you to go into are going to be late-stage PD patients. They may not see a whole lot of therapeutic effect here. One thing I didn't have a chance to touch on is that, that nigrostriatal pathway I talked about? Turns out, when you're diagnosed, within 5 years of diagnosis, so when you actually have clinical signs of it, you've already lost a lot of dopamine neurons. It's just that you have enough that can actually make up for those other ones. But when you get to a critical point is when you start to see the symptoms. That's when you're actually diagnosed. Within 5 years of that, roughly, all those fibers are gone. So those fibers actually turn out to be really good scaffolding for other fibers to grow on. So we think at least that if you treated a younger patient that didn't have as exacerbated disease, that you would get a more even effect and possibly even a quicker effect. you also would have that time I was talking about, that 2 or 3 years for the cells to mature and really start innervating. So I didn't mention this, but this is actually a good point. Just putting the cells in there is great and surviving is great, but we now think there's very strong evidence to suggest that those fibers that are coming out, that's actually the important part, that they're actually innervating and spreading that dopamine around. So our hypothesis is that I would rather have 1,000 neurons that were well integrated into the brain than 500,000 cells that were just kind of sitting there a little bit. And there's pretty good data to support that, some of which is from these types of cells in animal models, some of which is from humans. Still yet to be determined, but that is a great question because, and this is true with most diseases, you need to start with somebody. I'm going to use the eye as an example. These people were blind, so if they lost their vision, it wasn't like they were losing something. You didn't take something from them, right? They didn't have as much to lose. If you treat a late-stage patient and we find out that these things are firing weird, or they're causing, I'm trying to dumb this down just a touch just to make it a little bit easier to take, but they're basically causing your brain to fire in ways in which it shouldn't be firing. That's not good. That's going to be a side effect. And that was one of the side effects they saw with those fetal cells. So it's a great question you pose. It's one we don't know the answer, but we have to start with late, and then once you show it's safe, then you can get to effective. So for those who don't know, phase 1 studies are safety studies. Phase 2 are safety, efficacy in a slightly larger number of people. Phase 3 is when you have a ton of people, hundreds of people, if you will, in multiple centers, and everybody's doing the same thing. And so that is it repeatable around the world, around the country? To be determined. So great question, though. I think we had...
I was wondering if you're worried about an immune reaction to the cells.
Yeah.
How what level of suppressive would you compare to like a kidney transplant?
Yeah, great question. How worried am I? That used to keep me awake at night. So it turns out we have immune systems. We don't have the distinct ability to just take that gene out of us like we can in experimental animals. So the first strategy I told you about, the embryonic stem cell one, Blue Rock, they're going to use immunosuppression for 6 months. What we know from those fetal cell studies is that those cells worked for at least 6 months to a year when people used immunosuppression. It's actually a hotly debated topic, and the reason it's so important, and I didn't say this, was immunosuppression is extremely toxic. Its entire job is to kill cells, okay? It's not something you want to be on your whole life. So we are introducing a hole into the skull, and we're going down to the brain, so we're opening up the blood-brain barrier. So we do think we want to have that immunosuppression on for 3-6 months. There is actually data. I may actually have a slide hidden in here. Let me see if I have it. It's somewhere else. Doesn't matter. This group in Japan, Jun Takahashi's group, they show that you still get an immune reaction even if you take cells from the same monkey and put it back in the same monkey. There's still a small immune reaction. Okay, so I said in theory you shouldn't have a reaction. We don't know the answer to that. What we do think is that if you've had them for 6-12 months, and let's say you've had immunosuppression on board for 6 months or so, it's pretty safe. We've done our monkey studies out to a year with triple immunosuppression, which is, I actually designed that, and I spent a lot of time working with transplant surgeons trying to figure out exactly what we need, because there's no cocktail for brain transplantation. We don't know what you need. There are certain blockades we need to do in the immune system that we know for, say, liver versus the heart that are more important for others. So we actually designed this for something very similar to a low-grade whole organ transplant. And it turns out you actually need that. Because when we first started doing the studies, we just used what was in the literature from whole organ transplants, and it turns out we needed to increase tenfold the amount of drugs. The drugs are actually the most expensive part of the experiment, too. So the immune response is unbelievably important, and there's a lot of data out there to suggest that once the immune system starts attacking, you lose function. So again, this is a highly debated topic to this day with these companies that are sitting there going, What do we want to do? We'll see how it plays out. So,
Okay, I'm told that time is up. So sorry, a couple of you had more questions. And thank you very much to Dr. Wakeman.
thank you all.
Wonderful talk.